So when talk about the cells, the size will be very, very small. So that's why we have to view it using the microscope. Right? They are very, very small. So the smallest object of unaided human's eye can see is roughly about 0.1 millimeter long. Are you clear? 0.1 millimeter, you still will be able to see it as a dot, for example. Okay, but under the right conditions, some organisms like amoeba, proteus, human egg, the cell paramecium can be seen without using magnification, right? Proper state. Okay, if no, then we still can use a magnifying glass to see them, but again, they look very, very tiny unless we use a microscope. Okay, so in terms of this one highlight, so different type of cells, they definitely have different size, but generally eukaryotic cells is about 10 times the size of the prokaryotic cells, about 10 times the size of the prokaryotic cells. Plant cells generally are larger than the animal cells. If you compare, plant cells generally larger. Okay. So if you compare in terms of the size, like chicken egg, frog eggs, okay, we still can use the human eyes to see it. The neurons very long, you can see human eyes, okay? But after that, anything less than 1 millimeter, you might need to use a light microscope, okay? So for example, you can view epithelial cells, you can view the nucleus, you can use, okay, typical bacteria with a proper stain, okay? You will be able to view it, okay? Now, look at this, guys. Light microscope also can help us to see the chloroplast of mitochondria, but it won't be clear. Are you clear? It won't be clear, and sometimes you may not be able to see it. Chloroplast still can using the light microscope, but with the proper state. Mitochondria, most of the time, light microscope cannot. And some smallest bacteria, viruses, ribosome, okay, protein, amino acids, then you might need to use the electron microscope, okay? Electron microscope to see that structure. So later stage, we're going to look at what is different between the electron microscope and also the light microscope. Okay. So what are the factors limit the size? Why we do have this problem? Why the size cannot be bigger? Why humans, adult humans, why we are not unicellular but multicellular? Generally, we are limited by these 4 factors. So first, surface area to volume ratio. This is the one that we need to know. Okay, because when talk about surface area to volume ratio, when the surface area to volume ratio is high, so means that the transport, or we say that the diffusion will take place in a higher rate. Okay, so when something is big size, so surface area to volume ratio will be low. So that's why we cannot have a very, very big cells. Are you clear? Big cells. Okay. Now, and also we have to depend on the nucleus cytoplasmic ratio, fragility of the cell membrane, and also the mechanical structure. Now, what does this mean? Nucleus cytoplasmic, you cannot have a very, very small nucleus and very, very large cytoplasm. So they must maintain a certain ratio. Fragility of the cell membrane as well is something like balloon. You try to imagine that you continue to expand the balloon until the moment you're going to burst, correct or not? Right? Because the membrane cannot contain so much of the content. Okay? And also depend whether there are many mechanical structure that support the cells or not. For example, why plant cells generally bigger than the animal cells? Because plant cells, they have the cell wall. So with the presence of the cell wall, then the cells can actually grow slightly larger and elongate longer compared to the animal cells. Okay, so we have these 4 factors that limits the size of the cells. First, surface area to volume ratio. Surface area to volume ratio that we need to know in this case, basically it indicates the exposure. So when something is big, the surface area to volume ratio is small or low. When this ratio is low, so means the exposure is less. So means that we need to have transport system. Can I say that? Human, why we need to have the cardiovascular system? Because we cannot actually expose all our cells to the external surface. So try to imagine the cells inside our heart, the cells inside our lung. If let's say we don't have a transport system for them, then how they gain the oxygen? How they can remove the waste? Cannot, because humans is big size. So therefore, we need to have a special transport system because we are limited by the surface area to volume ratio. Okay. So again, out of these 4 factors, the only factors that we need to learn, next topics, deal with the diffusions and also the faceted diffusion. Then we're going to learn this surface area to volume ratio in detail, and we need to learn how to calculate as well. Okay, so with this, I stop the recording.